Today we had a very nice workshop and seminar on quality assurance and quality control in the clinical laboratory. It was a very interesting experience because people came from as far as Abu Dhabi and Alain to attend the conference. When we started with the idea of starting this conference, people told me that I would not even have 30 people to attend. But what was surprising was that we had 67 people who attended the conference and most of them stayed back till the end of the conference. So I'm surprised and pleased at the same time on the degree of involvement our technicians in our country have on quality in the medical laboratory. I see a great future for us.
Good morning to all of you. Ladies and gentlemen, it's such a pleasure seeing so many of you here in the very first seminar being conducted by the newly established Center for Advanced Biomedical Research and Innovation at our university. My name is Ravi. I'm from the Health Communications Department. On behalf of the founder president of this university is also the chairman of the board of governors, Mr. Tumbe Moideen. And being the very first event, I'm sure our provost, Professor Geeta Ashok-Raj, will be busy. She is in Hungary, so I'm sure she has sent her best wishes for this very first seminar. So on behalf of our leadership of the university and the academic leadership, and also on behalf of, I don't know if I can take the liberty to invite you all and welcome you all on behalf of Professor P.K. Menon, who is the Professor of Research and the Director of Center for Advanced Biomedical Research and Innovation. We are going to begin the scientific session very soon. The first session will be chaired by Professor Ishtiaq Shafi. And after 2 lectures, we'll have a small break where we will have our formal presentation ceremony, where we wish to honor all the speakers from within the center and also from outside who have agreed to be with us and share their expertise on this day. I see Mr. Austin Lal Jose here. Thank you so much, sir, for coming on please. Just as much as being
So we are going to start the first session, and there are 2 speakers. The first one is Professor P.K. Menon, who will be speaking on the analytical and post-analytical variables. And the second speaker is Professor Jayadevan Sridharan. He will speak on the applications of statistical methods and laboratory quality control. So this is, as Ravi said, is our first it's a continuous process. And we are never expert in this thing. We have to learn each and every day new things. And so going through his CV in India, Pune, in 1981, then he did his MD in medical microbiology and he has been a pathologist right from 1990 as a molecular. He has been a lab administrator and he joined GM medical journals and has conducted 80 projects in clinical trials. So,
It's actually very interesting. When I joined GMU rather, and I floated the idea of doing a quality control workshop for only laboratory professionals, people told me that nobody is going to come. So I'm really pleased today to see so many of quality management, where we run daily quality controls. This is our basic. So these are these various stages of quality. So here, for every test, if you have the funds, you run a quality control every day. Ideally, you should run it 2 times a day, one before the quality control, before the tests start, and you bracket it at the end of the day when the test ends. This is called bracketing. Bracketing is a very good practice because if the second bracketing run fails, you're able to rerun the whole batch, or you're required to rerun the whole batch. So this is quality control. Next level is quality assurance. Quality assurance comes as a system and as a process. Now, here what you're doing is, here you looked at the test, here you're looking at the process. Then you have a system which is a comprehensive and coordinated system to meet your quality objectives. So this is your 3rd level. The 4th level is quality improvement, where you have a formal process of monitoring the quality of your laboratory and increasing it or tightening your CV step by step. This is called quality improvement. And the ultimate, or what we all try to achieve, is something called total quality management, or TQM. TQM is the highest level of quality management in a laboratory or in any organization. Now, if you try to kind of align it with various regulatory requirements, CLIA and CAP does these first two levels. Your ISO 15189 covers the quality system also. 6 Sigma is quality improvement, and 6 Sigma is included in ISO 9001. But for laboratories, currently there is no ISO 9001. So laboratories, you've got ISO 15189, and the newer version of ISO 15189 also incorporates continual improvement. So I think this is the big picture which I wanted to give you before the start of the day. Equally important. The components of lab analytical quality are broadly the following. Loads which we send to the US to a lab in California, and this is a viral load sample, and look at the correlation. So this is the kind of group of laboratories within here, and we will try and exchange correlation samples and things like that on a regular basis, because that's essential. Comes reagent checks. So the kit should be shipped along with the calibrator. As I remarked previously, package, insert, review is essential, and SOP has to be reviewed ages of tests and persistent bias. Check QC rule violations, and all this comes with rigorous documentation, unfortunately, because any auditor who comes to your laboratory, unless the thing has been documented, will not believe that you have done it. So please better your comparison.
So it's best to go with CAP and then go past the ISO.
Either way is fine. Either way is fine. Ultimately, it get you to the same goal of TQM. Remember that first chart? I don't know if you missed that or not. The first chart which I showed you on TQM.
How many applications are there? In 45 minutes, it's not possible to finish all the applications. So I discussed with Dr. Main on what to improve, what content should be improved here. So as per his opinion, it's better to improve only basic things. That is the central tendency and dispersion, how these things are applicable in the laboratory, the control limit and laboratory, the controls, the quality control. So I'm going to include only basic things, that is the dispersion and the central tendency, how that is useful in the laboratory quality control. Okay, now the objective is only these things. So first you have to calculate how to calculate the central tendency and how to interpret and how it's applicable in the quality control, and how to calculate the control limits. If you take any parameter, any biochemical parameters or any parameter, there's a limit for that. How to calculate that limit? These are the only things we are going to deal with this particular. So the statistics will help you to find the accuracy and precision, and the calculating the control limits. Then, if you go further up, you can see to compare the new method with an existing method, how we can reach a new method is accurate while compared to the old. Exactly. It is the sum of the numbers divided by the number of observations. So, sum divided by number of observations. Clear? Okay. Now, the second measurement is the data. The first one, mean, median, mode, same. For the single values are there, so there is no mode. Which means some situation you cannot, the mode is not possible to find the precision. So we have to go for the next step from the each other one, each observation, other observation, how the spread out is. That is what we measure using the dispersion. There are two types of dispersions are there. Four. This is for main.
First 9.5, second one
with serum calcium value 9.6. mg per deciliter is in the acceptable limit. Abnormal. Then what about 99? And assume that instead of this 9.6, the value is 11.1. Then what? When you say when you say which, how much time is high?
Is the control limit same as confidence level?
This is confidence interval. Confidence interval. CS.
Level.
Make a syntax here and come back. Make a this is accurate.
Ravi, I hope you have had a very interesting first session. As I mentioned in the morning, we will have a very small formal presentation ceremony. This is basically as a token of our gratitude and wholehearted appreciation for those speakers who have accepted to be a part of this seminar, who are here with us sharing their expertise and experience. To begin with this brief presentation ceremony, we will have a recitation from the Holy Quran, and it's our proud privilege and honor to have the Dean, Admissions and Registers of the Gulf Medical University, Professor Muhammad Arifullah amongst us. Thank you for being with us during this session, sir. To welcome Professor Muhammad Arifullah, may I request Dr. Lehman to kindly greet him with the bouquet.
بالله من الشيطان الرجيم الله نور السماوات والأرض مثل نوره كمشكاة فيها مصباح المصباح في زجاجة الزجاجة كأنها كوكب دري يوقد من شجرة مباركة زيتونة لا شرقية.
It's been a singular privilege for me to be here and be a part of this evolutionary process for Gulf Medical University. We started developing a center for advanced biomedical research and innovation sometime in September last year. It is really a comprehensive facility which will give you advanced diagnostics, referral services and innovation in terms of drug development. So we are looking at a comprehensive structure where we have bioinformatics, which does all the drug design, molecular docking and similar studies. Then we have tissue culture and other facilities. The animal facility is coming up. We have an advanced molecular biology, the genomics laboratory. We have an advanced hematology laboratory. We have identified 4 areas where individuals in this country could benefit. That is diabetes, cardiac disease, allergies and congenital anomalies. So towards this we have set up a really state-of-the-art laboratory. Sometime during the course of the day, as you spend time here today,
Yes, congratulations to you. I'll hand over the stage to Professor P.K. Menon to continue with the scientific sessions.
Blessed with the attendance here on a weekend from morning till evening, and I want to thank for the invitation and a special thanks for the organizers, Dr. Menon, your team, for thinking of doing this, because actually day before I was in Beijing, yesterday I was in Shanghai. I spoke to the doctors there, why don't you do something like the QC seminar which is happening in GM. So starting on this note, what is a lab error and where can it occur? It can occur in any part of the testing cycle. Starting from ordering, patient preparations, error can be in any part, and it would lead to either a wrong result being reported. They would like to share, anybody would like to share. Were we talking about the pre-analytical phase so much 10 years back? So I'll go ahead, but this is the published data. Coming to the specimen, which part of this specimen workflow would be least prone to. What do you feel? You're working in the lab for, doctor, you're working in the lab for so many years. You tell me, which part of the... I'm Dr. Aparna Ahuja with BD Diagnostic. I head the medical affairs for them, in East Europe, Middle East and Africa. That's around 84 countries. And I am a speaker on pre-analytical quality today. And yesterday I was speaking in China on the same topic. And I'm very, my topic basically covered and made everybody realize what are the important issues in the pre-analytical phase of the lab and why quality is so important on the lab. As we know, only 5 to 10% of the cost of the hospital or the healthcare is contributed by the lab, but for the diagnosis, lab has about 70 to 80% of contribution. So even a small error in the lab leads to a major impact on the healthcare. So basis on that, and then it has also been documented that about 70% of the error happen in the pre-analytical phase of the lab. So it's very important to focus on that. I'm very, very happy that Gulf Medical University decided to start one of its kinds quality control seminar. And though I had to travel overnight and reach here to participate in this, but I didn't want to leave this opportunity, and I did mention because China I was there the last, so I did mention there also that they must try to do these kind of seminars, because it is, I think the thought that Dr. Menon and his team had in doing it was basically, it is us, the lab people. Like, I'm a bench level worker who's gone through past 20 years I've been working in labs, and quality is very, very close to my heart. That's why I'm now traveling everywhere and talking about the quality. So what I feel is that we are the people who can spread the quality awareness, because the eyes can see only what the mind knows. So this is one of the best steps that have been done to start the quality awareness journey in UAE, and I would look forward to many more events like this. When I entered the Gulf Medical University, I was amazed to see such a huge campus which has totally been dedicated to providing education. When I walk into the university, I see everybody working on a Saturday. I thought it was a weekend, but it seems here they work all the time. And when I come on the first floor, I see the lab, and I could not walk straight. I was just looking on the right, because it is beautifully made. I'm very happy that the management has decided to give so much of space to the lab, because they understand the quality of the lab reports are so important. And after I walked through, I met Dr. Menon, and I got to know that they are planning to have just about everything, atomic AS, LCMS, MS, MSMS. So I'm very happy. I think this is really going to be one of the reference centers here in this region. And thank you for making me part of the quality seminar, and I look forward to many such events. Also, I want to share that the people who are attending here, almost 50 people who are attending here, all of them are so involved in the seminar, and they are very, very well-aware people who would surely go back and contribute a lot to their own labs after attending this. Thank you.
Or we know, but some of the regulation tells us we have to use the quality control. The first one is ISO 15199. So we heard about the ISO 15199 from the very first presentation from Dr. Menon. Third-party quality control material should be considered either instead or in addition to any quality control material supplied by the reagent or instrument. It has to work in any of the instrument, and it has to give the most accurate result. The quality control body fluids. And it has to cover the clinically relevant intervals, because we have an unbiased assessment of the testing system. Says that control material needs to be different from the calibrated material, lot of variation. So the quality control should be give a long shelf life, at least only in an APL. They are categorizing the laboratory in 3 different: small, medium, and big. So it's for the
compact, no? Yeah, start. Action.
I am great honored to be here to part of Gulf Medical University, which started to the continual medical education event, and the first event is for the quality. So that means they are giving the most importance to
make or maintain the quality of the laboratory. And my topic was the external quality assurance program and the interlaboratory comparison program. So I have given the talk on the interlaboratory comparison program, which is the the largest interlaboratory comparison program is called UNITY. And the external quality control program, as well as the world largest or the world widely using the external quality control program from the Biomedical laboratories. And also I am very happy to be here with my Captain QC, and he has also seen the enthusiasm of the participants, and also he has visited the laboratory, which is a very good laboratory, and they are making this laboratory for their research and innovation for this part of the world. And also the participants, even it's a weekend, but the participants are a very huge number, and everybody is actively involving in this event. And thank you very much, Dr. Menon, who is taken the initiative, especially this part of the region, and I wish them all the best. Thank you.
You can do it in a batch run, or some lab will be doing a bracketing also, right? So you run quality control. What do you do with that? So you run quality control materials for a particular assay, say example of glucose. So you processed it. What do you do with that? Sorry?
We plot the results and see how it is fair in the graph.
Okay. So basically you interpret the results, right? So how would you interpret the results?
We have a set limit. We want to see near to the mean or at 1 SD or 2 SD.
Yes, basically. And what is Westgard rules? How to interpret?
First you have to collect your points.
Right. Then you standard deviation. Why it is written as 1, 3, yes. In your day-to-day continuous run, any one result value.
Hi, good afternoon. My name is Mariket Orbelio from Dubai, Neurospinal Hospital. I've been working there for 6 years as chief lab technician. We are here now in a seminar for lab quality control here in Biomedical University. The seminar is all about quality control for lab improvements, which is very informative, and all the speakers were excellent in their presentation.
Myself, I am Dr. Arvind Jain. I am coming from Alain. I work as laboratory director for Imperial College London Diabetes Center, managing their both centers, one in Alain as well as in Abu Dhabi. I heard about this quality control seminar recently through my friend, and thought of registering in it because it was being conducted by Gulf Medical University, a reputed university in UAE, and it was on a weekend, so I thought to attend it. And since today morning, I have been listening to various good speakers, and they have touched all the requisite topics related to the quality control issues in the laboratory. All the lectures have been very informative and educative. The hospitality has been very good, and we hope that we will have similar sessions on regular basis.
I'm Dr. Iman Dos. I am the lab director at Oasis Hospital Alain, and I was so impressed by the title of the seminar, which is held in Gulf Medical University in Ajman. And I came here, but I found that I am more challenged and surprised by the quality of the speakers and the depth they introduced or presented their topics, as well as the simplicity. I'm very happy that I attended this conference, and definitely if it will be repeated once more next year or whatever, I will attend, and I will inform the others to attend it. Thank you.